Hello, everyone, and welcome to this Diacetic event. This is an event that we have organized for the Blood Cancer Awareness Month. The title of this event is Chronic Myeloid Leukemia, the Essential Role of Molecular Testing as a Guide to Diagnosis and Treatment. My name is Antonio Capece, and this is the agenda of today's event. I will introduce you briefly the company, the Diacetic, and our platform for precision medicine. Then I leave the stage to Professor Soverini for her presentation. And at the end of the presentation, we have a Q&A session. During the Q&A session, everyone can ask questions you may have, and the expert will be more than happy to share her thoughts and experience related to the topic. Diacetic is a consulting and data analysis company specialized in precision medicine. We are committed to our goal that can be summarized in our motto, that is better testing, better treatment. We are striving for a future where all patients can receive appropriate treatment as soon as possible. We believe that labs need to be involved early in the precision medicine decision-making process. We have worked with different precision medicine drugs brought to the market with the world's most prominent pharmaceutical companies. We are a team that is globally present, spread over 53 countries. With this aim in mind, we have built the XRX, a platform conceived as a digital gateway that provides a space for all precision medicine stakeholders to collaborate with one another, as well as take advantage of our data insights. Laboratories, pharmaceutical companies, and diagnostic companies can all find benefits from being registered users of the XRX, and we expect this collaboration to have a positive impact in patients. We built the XRX diagnostic network to place laboratories at the center of the precision medicine ecosystem, as driven therapies will be not possible without their work. Those are some examples of what we offer to the labs through the XRX. The XRX collaboration, an opportunity for labs and other stakeholders to work together, for example, participating in ring studies or other scientific programs, as well as getting access to grants and sponsorships. Lab can also increase their visibilities to other precision medicine stakeholders, such as pharma diagnostic companies. Access to educational resources like this of today, lab talks, webinar, biomarker reports, or newsletter, and access to aggregated data by participating to our data strategies. This is conceived as something that the lab can benefit from, and such as membership is free of charge. If you are interested in this kind of activity of the lab offering of the Diacetic and the XRX, we would like to know more, please reach out to us. As we work towards a goal of better testing, better treatment, Diacetic wanted to highlight an organization that shares many of the same values. PathologyAlliance.com was established in 2001 with the mission to provide useful professional information to practicing pathologists and laboratory personnel in 15 seconds or less via a free and continuously updated textbook. This online textbook is a comprehensive and peer review written by pathologists for pathologists and touches on over 4,600 topics. They offer all pathologists a free editable webpage for life through filling out a survey, and they are currently looking for authors. For those interested in, you can send your CV and areas of interest to nat@pathogyalliance.com. We encourage everyone to visit their website and their offering. So, before to start this event, I would like to introduce you our speaker. It is a pleasure to have with us today Professor Soverini. Professor Soverini is Associate Professor of Hematology at the Department of Medical and Surgical Science at the University of Bologna, and she is part of the research group of the Institute of Hematology in the same university. Her research focuses on chronic myeloid leukemia and systemic mastocytosis. Professor Soverini is author of more than 180 publications in peer-reviewed journals, and she is a reviewer for a number of international journals, including Blood, Leukemia, Hematological, Clinical Cancer Research, and British Journal of Hematology, and sits in the editorial boards of Journal of Hematology and Oncology, Frontiers in Oncology and International Journal of Molecular Sciences. She is a member of the board of directors of the Italian Society of Experimental Hematology and a member of the European Hematology Association, the American Society of Hematology, and the Society of Hematological Oncology. She has served on the scientific program committee of HIHA and SOA Congress. So, thanks for your attention. It is a time to leave the stage to Professor Soverini. I hope you will enjoy this event. Thank you, Professor Soverini. I leave you the stage.
Dear colleagues, it is a great pleasure for me to be connected with so many of you today to talk about CML just one day after the World CML Day that, as you might know, was yesterday, September the 22nd, 9/22. So CML is the paradigmatic example of how the synergy between the lab and the clinic may be instrumental for therapeutic success. Actually, since the very dawn of the TKI era, molecular testing has proven to be essential for optimal patient management because molecular response soon became the gold standard for the evaluation of therapeutic efficacy, because key molecular response levels soon became important treatment milestones and endpoints, because regular monitoring of BCR-ABL transcript levels and kinetics are now key triggers for mutation testing and therapeutic reassessment, and because BCR-ABL kinase domain mutation testing may be an important guide to TKI selection in patients who are failing therapy. So those of you who have witnessed the clinical development of imatinib will remember when the first molecular response milestone was introduced in 2003 in the context of the IRIS study. That was major molecular response, the so-called safe haven for survival. And since then, we have come a long way with the introduction of the international scale for standardized assessment of major molecular response and all the cooperative efforts aimed to optimize and align the performances of greater and greater numbers of laboratories worldwide, so that nowadays, in many countries all over the world, each and every patient can be offered reliable and reproducible molecular response monitoring. Here you can see the international scale with the key molecular response level. You know that after major molecular response, additional MR milestones on the international scales have been consolidated with time, again with the joint efforts of the labs and the clinic. Molecular tools and protocols had to be enhanced to reliably assess the deeper levels of response, MR4, MR4.5, even MR5, and clinical studies demonstrated their prognostic relevance and their predictivity for TFR success. So nowadays, as you know, the achievement of definite molecular response milestones at given time points during therapy draws the ideal trajectory towards optimal response and the possibility to enter treatment-free remission and achieve what the late John Goldman used to call operational cure. So you can see here an early molecular response at 3 and 6 months, major molecular response at 12 months, and deep molecular response 12 months at 18 months, and beyond. So as our knowledge of how disease biology impacts on therapeutic efficacy continues to grow, and novel tools like next-generation sequencing and digital PCR are becoming more and more widely available, last year, together with my colleague, Professor Nick Cross, we thought it was time to produce a comprehensive and up-to-date document specifically addressed to laboratories and summarizing the current best practices for testing, at diagnosis, and during therapy. And the result of this effort that involved several European and non-European colleagues with a well-recognized and long-standing experience in CML biology and CML testing is this manuscript that was published in Leukemia at the end of last year. The manuscript is articulated in sections addressing molecular testing and diagnosis and molecular testing on therapy, both in terms of response monitoring and in terms of investigation of resistant mechanisms. So let's see together the main takeaways of this document. At diagnosis, karyotyping by conventional chromosome bending analysis, FISH, and PCR have a complementary role. Why? Because conventional karyotyping is important not only to detect the T922 translocation, but also to check for the presence of additional cytogenetic abnormalities that can be observed in up to 7% of our patients. The so-called major root additional cytogenetic abnormalities, major root ACAs, like trisomy 17 or isochromosome 17q, trisomy 19, trisomy 21, plus some minor root ACAs like deletion of chromosome 7q or monosomy 7, or 3q26 or 11q23 rearrangements, or again complex karyotypes, which means more than 3 different abnormalities, may signal for high-risk disease. On the other hand, qualitative PCR is important to assess transcript type, that in turn is essential for proper follow-up monitoring of our patient. So it should always be performed irrespective of whether conventional cytogenetic of FISH have already shown evidence of the Philadelphia chromosome. Why it is important to assess the transcript type? Because 98% of CML patients will harbor either the E13A2 or the E14A2 fusion transcripts. But as you can see here in the figure, there is a long tail of the so-called atypical transcripts that can be detected in the remaining 2% of our patients, resulting from breakpoints outside the major breakpoint cluster region on BCR or downstream of able exon 1. So it is essential to use a PCR strategy that detects both typical and atypical transcript prior to the start of therapy. Unfortunately, there are no CAE marked or FDA approved kits or systems that can be used to detect these rare isoforms. So you will probably be using some of the lab developed tests that have been published in the literature via multiplex RT-PCR. It is also important to be aware that there may be co-expression of transcripts, but this is due to alternative splicing. And if testing for rare variants is not available in the lab, then the diagnostic report should clearly state that the presence of BCR-ABL remains a possibility and that further testing in an appropriate reference lab should be performed. What else should we look for at diagnosis? As you probably know, it has recently been reported that mutations in cancer genes other than BCR-ABL are not only detectable in blast crisis, as we used to think, but also in a not negligible proportion of newly diagnosed CML patients. They have been reported in up to 30% of newly diagnosed chronic phase patients, and the most frequent mutations are in genes like ASXL1, RUNX1, DNMT3A, TET2, IKZF1, and so on. There have been recent reports suggesting that these mutations may be associated with inferior responses to imatinib and possibly even to nilotinib. And in those patients positive for mutations who still make it to achieve deep molecular response and become candidates for treatment-free remission, a report suggested that presence of these mutations are associated with increased risk of relapse upon treatment discontinuation. So a key question if these mutations are ready for diagnostic prime time. Well, there are several issues that still need to be solved. Technical issues, for example, whether we look for these mutations via panel sequencing or better directly via whole exome sequencing, and if in case of panel sequencing, what is the minimum set of genes to be screened for mutations? But also important clinical issues, clinical questions. So which genes truly harbor prognostic relevance, only XXL1 or all epigenetic regulators? Is any VEF clinically relevant? How to consider variant of unknown significance? Do we have sound alternative therapeutic algorithms for mutated patients? Well, currently the answer is no. And is there any TKI more active than others on given gene mutations or on mutations in general? So that said, within the ENLM panel, we decided to state that routine screening by myeloid NGS panel analysis or other NGS approaches are diagnosis is not disencouraged at all, but is generally considered to be research rather than a routine diagnostic requirement. So we do believe it's not inappropriate management of patients if we do not perform a myeloid panel diagnosis, although it may provide important information that will enable us in the near future to better stratify our patients. I know this is a busy slide, but you will see it in the manuscript. It's just a table summarizing all the tests that should be routinely performed or can be considered at the time of diagnosis in our CML patients. So, on therapy, serial monitoring of measurable residual disease should be performed by assessing BCR-ABL transcript levels in peripheral blood leukocytes by reverse transcription polymerase chain reaction. The frequency of monitoring should follow resources permitting that's specified by the current version of the ELN recommendations. So at present, every 3 months until MMR is achieved and confirmed, and then every 3 to 6 months. You know that there have been several studies over the past decade showing that digital PCR may be as robust and accurate, or even more robust and accurate than RT-qPCR. So, acknowledging this wealth of data, we stated in our recommendations that RT-dPCR is a valid alternative to RT-qPCR, and when optimized can provide more accurate results across a broad spectrum of disease levels, but may be of particular value at very low levels of disease, and I will say also in the setting of patients with atypical transcripts. The ratio between BCR-ABL transcript levels and the reference gene transcript levels should be expressed on the international scale as a percentage. You know that the international scale defines molecular response as log reduction in BCR-ABL expression from the iris standardized baseline. And this international scale can be adopted either using lab-specific conversion factors derived by sample exchange with an established reference laboratory. This is the way we used to go in the very past, or using nowadays more easy lab-specific conversion factors derived from secondary reference regions calibrated to the WHO reference panel, or validated diagnostic kits or devices calibrated again to the WHO reference panel. The problem is for patients with atypical transcripts. You know that for these patients, we cannot adopt the international scale, so these patients should be monitored by bespoke RT-qPCR or even better RT-dPCR assays, and results have to be expressed as individual molecular responses compared to the baseline level of that specific patient. So, in addition to BCR-ABL, the number of ABL1 or GAS-B or BCR reference gene transcripts must be determined for all samples, as you know, to gauge their quality and to estimate the level of MRD in samples for which BCR-ABL mRNA is undetectable. So, for example, undetectable BCR-ABL with 45,000 copies of ABL1 indicates MR4.5, but undetectable BCR-ABL with less than 10,000 copies of ABL1 indicate, as you know, that the sample is not evaluable for the molecular response because of poor RNA quality or some kind of technical failure. You know that BCR-ABL kinase domain mutations are currently the only actionable mechanism of resistance to TKIs. So many different mechanisms may underlie resistance in our patient, but at the end of the day, only mutations can give us information on how to best change the treatment when we want to switch. So the manuscript of the ELN laboratory recommendations also covers extensively the topic of BCR-ABL mutation testing, in particular in light of the wider and wider availability of next-generation sequencing. We thought it was important to provide indications on when and how to use NGS and how to interpret and not misinterpret or overinterpret NGS results. So, testing is indicated in case of failure and warning to first or second line therapy according to the current ELN clinical recommendations, and in case of relapse, which means loss of major molecular response after allogeneic stem cell transplantation, particularly if a mutation was detectable prior to transplant. In contrast, testing is not indicated at diagnosis before the start of any TKI treatment, at failure after TFR, except for those rare cases not achieving again major molecular response within 3 to 6 months after restarting TKI, which is indeed quite rare. And in general, in all patients who have a response, like major molecular response or better, and if both for technical reasons and for clinical reasons, technically because it's not feasible, not even with NGS, clinically because it does not make sense. How to perform BCR-ABL kinase domain mutation testing? Well, targeted NGS, whenever available or accessible, is the recommended method for TKD mutation testing, provided that a reliable and validated assay has been implemented. The recommended input material is RNA from peripheral blood or bone marrow leukocytes. And it is important to bear in mind that analysis of genomic DNA is not recommended for routine analysis. So please avoid using myeloid panels for BCR-ABL kinase domain mutation testing. Digital PCR may be considered as a first-level screening test for detection of specific mutations of interest, like, for example, the T315I, when rapid TKI switch is needed. However, we underlined in our document that the use of Sanger sequencing is acceptable whenever NGS is not available or accessible and does not represent an inappropriate management of patients. Here you can see a table summarizing the mutations that impact on TKI selection once they are detected in a resistant patients. We also tried to provide a provisional list of mutations conferring resistance to asciminib, but list based on merged information from in vitro studies and the first clinical trials that are still ongoing with this TKI, bearing in mind that this list is still very, very much provisional, and we will need more clinical experience with asciminib to have a full picture of all the mutations that will confer resistance to this novel allosteric tyrosine kinase inhibitors. And most importantly, how to use NGS BCR-ABL mutation results. Here in this slide, you can see a useful algorithm for clinical decision making based on NGS results. So, if a mutation is detectable by Sanger sequencing, or if a mutation is detectable by NGS in greater than 15% of the transcripts, so we have a big mutated clone, then we need to ask ourselves whether this mutation is resistant to the ongoing TKI. If yes, it's definitely time to change the therapy. If the mutation is detected by NGS in less than 15% of the transcripts, which means we have detected the so-called low-level mutation, then again the question is if the mutation is resistant to the ongoing TKI, and if yes, if the risk of continuing the ongoing TKI treatment is much greater than the benefits, then we should change the therapy. If no, we can better adopt a watch and wait approach and repeat testing in a subsequent sample. Clinically relevant mutations always get selected in subsequent samples if the TKI is not changed. And here again, a second table summarizing the recommended tests for monitoring or investigated CML patients on treatment. And that was my last slide, and I would like to thank all the colleagues who contributed to creating this document that I think it will be an important tool for all colleagues working in the laboratory and dealing with CML samples. I would like to thank the ELN and all of you for your attention.
Thank you, Professor Soverini, for your really detailed presentation and full of really important information about CML and how to manage. So we are opening now the Q&A session. So waiting for a question from the audience. If you wanna, I have a couple of questions for you.
Mm-hmm.
So the first one is why myeloid NGS panels are not suitable for BCR-ABL mutation testing?
Yes, this is indeed a question I got from several colleagues, and I didn't have the time to specify during my presentation. Well, nearly all myeloid panels indeed include ABL1 as the target genes, but there are two important problems. First and foremost, the use of genomic DNA does not allow the selective amplification and sequencing of the translocated ABL1. So the normal ABL1 and the translocated ABL1 will be sequenced all together with an important loss in sensitivity, given that mutant ABL1 molecules will be greatly diluted. And the second, but not less important, some commercial panels do not cover all the ABL1 exons that need to be sequenced. It is mandatory to sequence the region encompassing exon 4 to exon 10, while some panels are limited to ABL1 exons up to 7. So again, this is another important thing to be borne in mind.
Okay, thank you for your answer. So we have an answer from the audience. Is Dr. Anthony Abaldi. Many thanks for such a wonderful brief. What if at diagnosis lymphocyte levels are alarmingly high? Does that warrant TKD testing at diagnosis?
No, I would say no, because even in such case, there are no data suggesting that there may be mutations already detectable at diagnosis, and at least with the tools we currently have. Most probably because in the absence of any selective pressure from TKI treatment, these mutant clones are generally less favored as compared to the wild type BCR-ABL, so they are not taking over wild type BCR-ABL. So really, there are no data suggesting that we should look for mutations in this setting. I will expect a wild type result even with NGS.
Okay, thank you. Another question is the following one. So you mentioned in your presentation digital PCR that is now acknowledged as a valid alternative to real-time quantitative PCR. But are we ready for routine use of this PCR in CML?
Well, I think there are settings in which digital PCR may bring immediate benefits, like, as I mentioned, the setting of patients with atypical BCR-ABL transcripts, because, as you know, these patients are currently orphan of quantitative MRD monitoring whatsoever. So, for example, they do not have access to treatment-free remission, because we would not be able to check whether they relapse or not. So in this setting, it can be very useful because we do not need any kind of standard curve. It's much more straightforward to quantitate these atypical transcripts. In other settings, I think there is still work to do before routinely adopting digital PCR. Because for example, we do not have comparative data regarding the performances of the different platforms or of the different kits. We do not have robust and reproducible cutoffs to stratify TFR candidates, for example. So even though digital PCR has been shown to be potentially useful to better stratify TFR candidates, there is no reproducibility across the different studies in terms of the criteria we can use, and so on. So I think there is still a lot of work to do in this sense.
Thank you. We have a question from Dr. Madhu Diwan. Do you get mixed CML with lymphoid transformation?
Mixed CML. Sorry, I didn't mix the
Yes,
CML.
mixed CML with lymphoid transformation.
I'm not sure if I got it correctly. So myeloid and lymphoid plus crisis altogether, or?
We will ask more detail to Dr. Diwan. So if you,
Okay,
Dr.
I'm
Diwan,
sorry.
want to, yeah,
Yeah, if you can clarify.
yeah. Dr. Diwan, if you want to clarify, oh, it is here. Please highlight refractory CML on going into plasma blast transformation, treated sickle cell.
Okay. No, this is really something I've never experienced in my professional career. I'm sorry, I cannot comment on that.
Okay, so, okay, yes, please, blast crisis. Okay, so we wait for other question if we have. Okay, so waiting for other questions. What do you think about the use of CAR T cells on CAR NK cells for the future, for in this case for blood cancer and solid tumor? If you have an idea, or if you think that could be really useful to mix this for the future.
Well, yeah, of course, the chapter of the big chapter of CAR T and more recently CAR NK is really an exciting story that holds the promise to become. It's already a great breakthrough, and also promise to become even more a breakthrough in the future. As the technology of creating these CAR T and current cases is evolving. If you consider all the generations of CAR T from the very beginning to the molecule, to the drugs we are using now, it's really incredible what we have done so far, and I think that it's really changing the way we are treating some diseases. This is not something. This is something that is far away from CML, but in other disease settings is really incredible. There are still, as you know, some issues to be solved, but clinically speaking. But I think that it will be the future in several instances.
Another question from Dr. Diwan. So, treated sickle cell anemia manifesting CML, is it refractory, resistant to treatment? Why is it so?
I think it's difficult. I mean, resistance to treatment, it's difficult to investigate the problem of resistance with the tools we have nowadays, at least routinely speaking. So I think that except for mutation testing, we do not have many other ways to investigate this problem, except for just changing the treatment and seeing what happens. It's very empirical, but this is the only way we have.
So there is a lot of way or roads to run again for the future. So thanks for the answer. So if there are other questions, otherwise I think that we can say thanks to everyone. So if you have a question, even in the next day, so please send us in order that we can forward to Professor Soverini and we can answer you later. So thank you all for your participation and for attention. And thanks to you, Professor Soverini, for the presentation. Really interesting, as I said before. So thanks again, and speak to you soon. Oh, there is another comment for you, Professor. Fantastic presentation. That's perfect. So thanks again to everyone, and have a good continuation of the day.